Hello, everyone, and welcome to today's webinar, On the Right Track, Building the Neural Circuitry for Feeding and Swallowing, presented by Dr. Zahra Motahari, Postdoctoral Scientist, George Washington University. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Microsystems. To learn more about our sponsor, please visit leicamicrosystems.com. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the left side of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Zahra Motahari. For a complete biography on Dr. Motahari, please see the Biography tab at the top of your screen. Dr. Motahari, you may now begin your presentation.
Thank you. Thank you for the introduction, and thank you, Leica, for inviting me to speak and talk about my project. I'm very excited. My name is Zahra, and today I'm going to talk about the project that I'm working on for like around 2 years, and that would be focused on the neural circuitry for feeding and swallowing. But let me start off by briefly explaining why it's important. In humans, there is a genetic disorder called 22q11 deletion syndrome, which is caused by a microdeletion on the proximal portion of the long or Q arm of chromosome 22. 22q11 deletion syndrome is very common. It occurs in approximately 1 in 4,000 live births. And deleted individuals show complex and variable psychiatric phenotypes such as schizophrenia, ADHD, autism, learning disabilities, as well as physical phenotypes. The physical symptoms are very broad and include developmental delay, congenital heart defects, craniofacial anomalies, hearing loss, musculoskeletal anomalies, absent or small thymus and parathyroid glands, compromised immune system, and dysphagia. So what is dysphagia? Dysphagia is feeding and swallowing difficulties. It occurs in 35 to 80% of children with neurodevelopmental disorders. It results in poor nutritional status and increased risk of nasal, middle ear, and respiratory infections. And unfortunately, the causes are unknown. If you look at the feeding and swallowing process itself, it relies on rapid coordination of the, oh great, I can use the mouse here. So it relies on rapid coordination of the muscles and structure in the mouth, throat, and esophagus. So this apparatus is controlled by a neural circuitry composed of motor neurons from the hindbrain and peripheral cranial sensory neurons. The motor command includes the maxillary and the mandibular branch of trigeminal nerve or cranial nerve 5 for jaw closing, the facial nerve 7 for jaw opening and movement of facial muscles, cranial nerve 9 and 10 for pharyngeal and laryngeal muscles, and cranial nerve 12 or hypoglossal for tongue muscles. And if you look at the mouse, you can see that the structure and pattern of these cranial nerves are very conserved, which is great because now we can use a mouse as a model system to study dysphagia. But for today, I'm going to just talk about the cranial nerve 5. And why cranial nerve 5? Why is important? Let me start off by just go a little bit in details about cranial nerve 5. Cranial nerve 5 is responsible for sensation in the face and the motor functions such as biting and chewing. It has 3 major branches: ophthalmic, maxillary, and mandibular branch. The ophthalmic and maxillary nerves are purely sensory, whereas the mandibular branch supplies motor as well as sensory functions. The sensory functions of cranial nerve 5 is to provide tactile, proprioceptive, and nociceptive to the face and mouth. So just a little bit more details. Proprioceptive is a sense of relative position. For example, the position of the food in the mouth is sensed by these receptors. And nociceptive can be divided into 3 categories: chemical sensation, such as chili powder in the mouth; mechanical sensation, like cutting and crushing; and of course, thermal sensation for the sensation of the heat in the mouth. And the motor command control the movement of 8 muscles, including the 4 muscles of mastication. And together, these sensory and motor neurons coordinate the movement of the muscles and structure in oropharyngeal apparatus, and of course, with the help of other cranial nerves. So we have a mouse model for 22q11 deletion syndrome, which is called large cell. The first thing we did, we examined whether or not large cell pups can exhibit similar defining feature of dysphagia. And so we found that, like humans, large cell pups fail to gain weight. They also have milk aspiration into the respiratory passages. We also saw sign of infections. So what I'm showing here is an example of milk aspiration in P3, P4 larger pups. Fluorescent dye added to milk just for the illustration purpose. And so you can see that in Y-type pups, when milk is swallowed, is nothing left in the mouth and the esophagus. However, in large cell pups, milk present in the mouth and esophagus, and also is present in nasal cavity, which is one of the symptoms of dysphagia. So these results actually confirm our suspicion that this mouse model can be used as a mouse model for studying dysphagia. So that was great. And then we look at the development of cranial nerve 5 in this mouse. So what we found was that in large cell animals, also the development of cranial nerve 5 is disrupted as early as embryonic day 10-5. So this is a larger image. You can see that the nerves are sparse and less branched. Also, the complexity and length of both maxillary and mandibular branches are significantly diminished. In other words, we did see an abnormal gross morphogenesis in mutant animals. Although these findings are very informative, but they don't show whether or not the neural circuits that innervate the oropharyngeal structures are also disrupted. In fact, the development of neural circuitry depends on 3 major processes: cell fate specification, synapse formation, and axonal guidance. Our lab has been focused on 2 of these processes, cell-phase specification and axonal guidance. And we have a strong evidence that in mutant animals, the balance between cell proliferation and differentiation is significantly disrupted. But I'm not going to talk about that story today. I will just focus on axon pathfinding. In order to investigate axonal growth, we needed to study them in their native 3D structure, which was not accessible by conventional serial sections. So what we did, we labeled axons with a neural tracer and then utilized confocal microscope to look at the axonal trajectories and morphology. We use biosighting as our neural tracer. Here is an example of 11.5 embryo injected with biosighting. Roughly around 20 to 40 nanometer biosighting is injected in the branchial arch 1B, which makes the lower jaw, with a glass macropipette under microscopic observation. After injection, embryos were transferred to cell culture media and incubated for roughly about an hour and a half at 37. Then day 6, clear and processed for 3D imaging. Here I'm going to show you a video of the wild-type embryo injected with biosighting at 11.5. One of the qualities of biosighting is that it can travel in both directions, anterograde and retrograde. So after injection, biosighting travels through the axons retrogradely and label the cell body of the sensory neurons in the trigeminal and motor neurons in the hindbrain. You can see that the axons are labeled very nicely, and you can actually track them to the injection site. This is a Thai image of the embryo. It's about 350, 400 micron deep. And the image, again, is the injection site, trigeminal, and the hindbrain. And it takes about 17, 18 hours to be completed. And you can see that the cell bodies, I'm not sure if you can actually notice, but they are located in different planes. For example, the sensory neurons are located more to the surface, and the motor neurons, they are located deeper in the tissue. So, I'm sorry. Okay. So here what I'm showing is a snapshot of, again, the trigeminal and the hindbrain. And again, I wanted just to emphasize that the sensory neurons, the cell body of the sensory neurons are located in the trigeminal ganglion, and the motor neurons are located in the hindbrain. Another thing is that the sensory axons going to the hindbrain and bifurcate very nicely in the hindbrain and going directly toward the target. Here is again another example of injected embryo, and you can see that the axons label very nicely from the injection site, and the dye goes to the cell body of sensory neurons, and you can actually see the axons going to the hindbrain. So by now we developed a technique, and we're ready to look at the axonal growth and pathfinding. So the first thing that we asked was whether or not axons going to the correct target. So to answer that question, we injected sensory neurons in the trigeminal ganglion with biosighting conjugated to alexaflor 594 and 488, and observed the axonal trajectories into the branchial arch 1A, which makes the upper jaw, and arch B, which makes the lower jaw. This is an example of the wild-type embryo. injected, and you can see that the sensory neurons in the anterior portion of the trigeminal ganglion going directly to the arch 1A, and the one located in the posterior portion going to branchial arch 1B. In other words, they are very organized. And we repeat the experiment twice and compared 20 wild type and largely mutant, and in fact, we didn't see any significant difference between large-cell and wild type. In other words, even in mutant animal, the axons could find the target and go to the target directly. So there was a difference here. Then we look at the axon morphology. So to do that, after taking 3D images, I use a software to track the axons. And to be precise, I actually went through each Z-stack plane by plane and manually traced the axons, and then use the software to connect the dots and give me a 3D image. This is an image of the tracked axons. There are 4 key points here that we have to pay attention to. First, you can see that the cell bodies of the sensory neurons located in trigeminal ganglion and motor neurons are in the hindbrain. They both are going to the correct branchial arch. Sensory axons bifurcate in the hindbrain very nicely, and also motor axons coming out of the cell body and going directly to the target. So we repeat the experiment in mutants in large cell embryo, and we did see that like wild type, the cell bodies are located in the correct position, and both of them actually, both sensory and motor neurons, going to the correct branchial arch. But we did see 4 abnormal phenotypes, one of which is, we call it looping. We have, here is examples of looping. This is a larger image of these purple neurons here. You can see a looping. We have a looping here in the hindbrain, this is a larger image. This is what I call data looping because it's just like 2 loops together. And here is another example of data looping in the hindbrain. I should mention that the looping is not specific to the hindbrain. We do see looping in the hindbrain as well as in the periphery. The second phenotype is what we call it sprouting. So if you remember, wild type axons going to the hindbrain and bifurcate very nicely and going to the target, but what we see here is like it seems that the axons are in the navigation mode when they try to sense the environment, which is abnormal in this location. So we call this morphology sprouting. The third morphology was what we call misrouting. You can see that it seems that the motor axons here is kind of lost, going to the wrong direction, and somehow corrected itself and then coming back up and going to the right direction. And the fourth one was branching. So sometimes close to the end of the axons, or sometimes even in the middle of the axons, I saw that the axons branch or make a fork, and then from one cell body there was 2 axons coming here going to the target, which was abnormal. This is a summary table of the results out of 13 wild-type embryo and 56 axons. I saw that every one of them going to the correct branchial arch. I did see 46% looping in wild type, and also I did see abnormal phenotypes with low frequency, 1.7% branching, 5% misrouting, and 0% sprouting. In mutant animal, out of 15 embryo and 70 axon analyzed, again, they are going to the correct branchial arch. 55% of them make looping. But if you look at the other phenotype, the frequency of other phenotype was much higher in larger animals. 11% branching compared to 1%, 17% misrouting compared to 5%, and 1.4% sprouting. If you put everything in a table together just to compare, you can see that it seems that there is no difference in looping. They are kind of very similar, but you can see that the frequency of abnormal phenotype is significantly higher in large-scale animals compared to those in wild type. And this is very much significant. But so even though this looping, I was thinking about it quite some time, and it kind of bothered me in terms of why we do see the same frequency. And I realized that I should mention that the way that I score this phenotype here, when I saw one loop, I scored it as a looping phenotype. But then I realized that there are more looping in large scale. So then I counted each loop, and then this is a summary of the results. Very interestingly, so the y-axis shows the percent of the axons, and x shows the number of loops. You can see that in wild type, more than 60% of the cases, they have just showing one loop, and around 20% two loops, and very few showing three loops. However, in large scale, it was the opposite way. So around 20% showing one loop, and then most of them, like 30%, two loops, and I also saw three loops and even more than 3 loops. Sometimes I did see 10 loops in one axon. And that difference was significant. We do see more looping in mutant axons. And also, I looked at the number of phenotypes per axons. So, and the result is summarized here. The y-axis shows the number of phenotypes, and x shows each condition, and each dot shows represents one axon. So you can see that 30 axons, wild-type axons, they don't show any phenotype. I do see one phenotype, which was mostly looping in 23 axons, and only 3 wild-type axons show 2 phenotypes. Whereas in large scale, 25 show no phenotype, 32 had 1 phenotype, 11 axons show 2 phenotypes, which was significantly more than 3 in wild type, and also I had 2 axons that show 3 phenotypes. Again, the difference was very significant. So just to summarize this part, we found that in wild type and large scale animals, both sensory and motor neurons directly project to the correct branchial arch. Large scale cranial nerve axons, however, exhibit abnormal phenotypes such as looping, branching, misrouting, and sprouting. So these phenotypes are rarely documented in vivo in the way that we have, and so our novel approach allows us to observe this phenotype with cellular precision. So then the next question that we asked was, what is the mechanism for this disruption and this abnormal morphology? If you look at the hindbrain development, one of the signaling molecules that establishes the anterior-posterior polarity is retinoic acid. Here is in situ hybridization of E95 hindbrain with CYP26B1 probe, which is a retinoic acid regulator catabolic enzyme. You can see that in wild type, retinoic acid is mostly expressed in the rhombomer 5 and 6. In large scale embryo, however, retinoic acid expression is expanded toward the anterior rhombomer, suggesting that the identity of the cells in the anterior portion of the hindbrain has been disrupted. This is very, very important because the early patterning of the hindbrain is crucial for not only motor neuron specification, but also neural drive, peripheral sensory neurons, as well as branchial arch differentiation. If you remember, I said that the cell fate specification and axonal growth, which is guided by the environmental cues, which in this case is environmental cues in the branchial arches, are very, very important for establishing the neural circuitry. Interestingly, by genetically lowering the expression of retinoic acid in the hindbrain, we were able to restore wild-type cranial nerve phenotype in large cell mice. In other words, by restoring the identity of the cells in the anterior hindbrain, we rescue the axonal growth and cranial nerve phenotype, or should say cranial nerve 5 phenotype. Now the question is whether or not we can rescue the abnormal axon morphology at cellular level. So to answer that question, again, I injected biocidin into the branchial arch 1B, let it sit for an hour and a half, and then 6 clear and process the embryo for 3D imaging. And this is a summary of the results in rescue animals out of 11 embryos and 66 analyzed axons. Again, everybody going to the correct branchial arch. I did see 53% looping, which was very close to wild type, but I did see abnormal phenotype, which less frequency in rescue animal. I did see 3% branching compared to 11% in large cell, 4.5% shrouding compared to 17% in mutant animal, and 0% shrouding compared to 1.4%. Again, if you want to summarize, you can see clearly here that the frequency of the abnormal phenotype is very similar to those in wild type. And in fact, there was no significant difference between wild type and rescue animals. And again, if you look at the number of phenotypes per axon, again, the y-axis shows the number of phenotypes, and we have each condition here. You can see that the rescue are very similar to wild type. We had 30 axons with no phenotype, 32 axons with only one phenotype, and 4 axons with 2 phenotypes, which was very close to 3 axons in wild type. So the conclusion is that, yes, by rescuing the identity of the cells in the hindbrain, we were able to not only restore the cranial nerve phenotype and growth, but also we were able to rescue the axonal morphology in these animals. And just to summarize, we show that in large cell, the anterior-posterior patterning of the hindbrain is altered. Also in mutant, the cranial nerve 5 axonal growth and path binding are disrupted. We found that cranial nerve 5 phenotype in mutant animals can be rescued by genetically reducing the level of retinoic acid in the hindbrain. And finally, the 3D imaging help us to resolve compromised axon path binding in mutant animals that might otherwise have been missed using conventional techniques. And with that, I would like to thank people who helped me in this project. I would like to thank my advisor, Dr. LaMantia and Dr. Moody, Dr. Popretilov, the Director of Nanofabrication and Imaging Center, and Dr. Maynard for technical support, and of course, everybody in the lab. I would like to thank the Department of Anatomy and Cell Biology, the Department of Pharmacology and Physiology, and also the Nanofabrication and Imaging Center, and of course, the funding resources. And thank you very much.
Thank you, Dr. Motahari, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Answer Question box located on the far left of your screen. We'll answer as many questions as we have time for. Okay, let's get started. Our first question is, Dr. Motahari, what is the specimen thickness limit for imaging in 3D with sufficient resolution to do single axon process tracing?
Well, it really depends on the objective that you're using, and of course the method of the clearing. In my case, I use BABB, benzyl alcohol benzyl benzoate, and I use oil immersion 20x. And by using that specific objective, I have like the maximum of 450 micron working distance. But again, if you're using clarity and then use the water immersion objective, you have much more working distance, for example, 3-4 millimeter. Again, it depends on your objective.
Okay, our next question, how much depth can you measure from imaging?
How much depth in terms of, again, it depends on your objective. If you using water immersion objective, you can cover up to 5-6 millimeter.
Now, are there any alternate? Are there any alternate clearing methods other than BABB?
I mean, of course, there are a list of other reagents that you can use, and it really depends on what your question is or what tissue you try to clear. And so based on that, you can use different methods or optimize based on your own condition. For example, one of the early tissue clearing methods was BABB and DISCO, which is based on organic solvent, but then there are other bunch of chemicals came along, which are more like a hydrophilic chemical, urea, for example. If I want to give you a couple of examples, scale, CDB, clear. And another one that actually people have started using it very much is clarity, which uses embedding the tissue into hydrogel polymer, and then you can use oil. Water immersion objective. There are a bunch of them, and this is just some of them that I mentioned.
Have you tried whole mount antibody labeling combined with biocytin tracing?
That's a good question. Not really. We thought about it. It's very cool if, for example, if you want to look at, for example, cranial nerve 5 and see which one going to the branchial arch. You can do, for example, TUJ1 antibody staining combined with biocyting, but we didn't do it. That's a good way of doing things, but that was not our concern, and no, we didn't try that.
Okay, our next question. In one of the imaging you used 2 dyes, and it appears as some of the neurons are double labeled. Is it because of the properties of biocytin that can cross through gap junctions, or because these neurons send branches to both locations?
Well, actually, that's a very, very good question. And to be honest, we have some idea, but we are not sure. One of the, I don't know, maybe drawback with biocytin, it leaks. So even though you inject to the anterior portion, it leaks a little bit going through the cell body in the posterior portion. Double labeling could be the leaking of the biocytin, and this is what I'm willing to accept. But rather than getting like branches in two branchial arch, we didn't see that much evidence to support that they getting two label axons, because it was just cell body that were labeled, not the axons. I would guess that it was just some leaking going through the cell body at the injection site.
Our next question: could sparse recombination brain-bow approaches be used with this technique to resolve multiple axon processes cells in one single animal and increase overall N's for measurement?
Well, I mean, let me explain with what brain-bow is for the people who don't know. Brain-bow is a process by which individual neurons in the brain can be distinguished from neighboring neurons, and then it has been developed in Harvard University. And the way that it's been done, they're using Cre-Lox recombination in order to put a specific fluorescent under a specific promoter, then different cell type express different dye, which is very nice. In terms of increasing the number of N, and I assume N meaning the number of axons, I don't think so, because the number of axons is just the one that is labeled with biocidin. But the cool thing, if we combine brain-bow with biocidin, then we can actually distinguish, for example, if any specific motor neurons in the hindbrains is more disrupted than the others. And this phenotype, because by the way that I've done, I have no idea which type of motor neurons in the hindbrain disrupted. Is it all of them? Is it some of them? And that method, the combination of brain-bow with biocidin, could actually answer that part of the question, that if any specific motor neurons are being disrupted or more affected by this deletion. That's a good question.
Thank you, Dr. Motahari. Let's see.
Sure.
Let's see. Okay, it looks like we have time for one more. Let's go with this one. Do you think the looping phenomenon is a normal process that is unregulated in the deletion model?
It could be. Well, to be honest, we are not sure. We don't know if looping has any function in like axonal morphology, or what it do anything. So first of all, we don't know that part. But I found a literature in the brain that author suggests that the looping could be a sign of degeneration in the neurons in the hindbrain. We were not sure if this is the same thing that we are seeing here, but it could be, and we don't know. I mean, this is just kind of a speculation. And in terms of what it does in wild-type axons, we don't know. I don't know if it has a specific function, to be honest, in wild-type axons.
Thank you, Dr. Motahari. Do you have any final comments for our audience?
The final comments is, I think the method that we are used could actually help us to answer the most basic questions. Again, if you wanted to section through the axons or section through the hindbrain, we probably could not get the good idea of the morphology and what's going on in the axonal pathway. Just using confocal microscopy just give us the power to solve this problem at the cellular resolution, which was actually very cool.
Thank you, Dr. Motahari. Thank you so much for your time today and your important research.
Thank you for having me.
Absolutely. And we would also like to thank LabRoots and specifically our sponsor, Lycra Microsystems, for underwriting today's educational webcast. Now, before we go, I would like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand through April 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thanks for joining us. Until next time. Goodbye.
Okay, thank you.
Hmm.